Divide the lecture into 3 parts. The first part, very simply, I'm going to show you the epidemiology. I'm going to show you that many groups over the years, the last decade, have shown that there is an increased risk in large cohorts of individuals. I'm going to show you the animal models that we use to try and study the mechanisms that are involved. So I don't like to say bad things about people, but epidemiologists do these large cohorts, they do these studies, and they show a connection between 2 things. But it's soft data. They don't actually show you what the cause is. They just tell you there's an association. And so it's important in terms of understanding the mechanisms so you can consider drug targets or ways to prevent the diseases, etc. So that was the prime reason for doing these experiments. Let me show you some of the epidemiology. Here's a study from the New England, which is everybody's favorite journal. And what they showed, and this is a cohort from the US, but it's been replicated in many countries around the world, that the mortality from cancer is increased if you're obese. And the list of cancers involved are all of the above, except for one. The one in men is prostate cancer. All of these cancers that are listed in women and men are more common in obese individuals and more common if you have type 2 diabetes, except for prostate cancer. Men who are obese seem to be protected from prostate cancer. But if they have prostate cancer, they're more likely to die. So you have to separate out the incidence. And the study was basically they took men who had a certain Gleason score, had total prostatectomies, and then divided them into 2 groups. And the obese men, post-op, had a higher mortality than the thin men who had the same type of prostate cancer. But other than that, everything holds up. The internists and gynecologists will remember for many years we've known about endometrial cancer, uterine cancer in women is increased manyfold if they're obese. And that is held up in many of these studies. And this just shows you how it's ordered. Basically, the increased BMI, the more obese in men from the US, but around the world, liver cancer, pancreatic cancer, etc. All the cancers are basically 1.5, 2-fold increased risk. And in women, much the same. You'll see uterus 6-fold, but kidneys increase, cervical, etc., pancreas again. So there's no question from all these studies that there seems to be an indication that obese individuals have more risk of cancer and higher mortality. So one of the questions was, if this is true, can we treat the obesity and actually prevent cancer death, for example? There are 2 studies in the New England. This is the earlier one from Utah, and there's a recent one from Sweden. And basically what they did is they took 10,000 patients who had gastric bypass surgery for obesity. And they compared them with 10,000 obese individuals who were treated by endocrinologists and internists. And you can imagine, the surgeons did a better job than we did as endocrinologists. So you had a control group that didn't lose very much weight, and you had a control group that lost a lot of weight. And if you look at mortality, you'll see that the mortality was down by 50%, from 61 to 37 in the treated surgical group. Cardiovascular disease was down significantly. They developed more diabetes in the control group, less in the group that was now much thinner. But what was sort of hidden in the data is that cancer mortality was reduced by 2/3 in those that underwent bypass surgery. So the more obese you are, the more likely you are to develop and die from cancer. And if we treat the obesity, you're less likely to die from cancer. So that seems like a good sort of setup for the following experiments. The patients that I deal with are shown on the next slide. They're much easier to deal with than the patients that everybody else here deals with. Basically, what we did is we took mice and we made them obese. And the way that we did this, we started at the NIH, so we used the NIH cafeteria diet. And we moved to Mount Sinai, and we continued with the Mount Sinai cafeteria diet, and they're identical. They're all high in sucrose and high in fat, and so very quickly you can see the regular chow mice remain normal in growth, and the bottom you can see the cafeteria diet mice who become obese. And that's shown here. In the white bar, you'll see their starting body weight, males on the left, females on the right. This is in the gray bars what they would grow to over the few weeks of the treatment and the regular chow, and the black bar is obviously when they're on the high-fat diet, they become very obese. And you'll see that the percent of fat goes from 10% to 30%. So no question that this is a great model that was developed by many other groups. What you'll also notice for those involved in obesity research is that the leptin went up very high. That's also known. The more fat cells you have and you're obese, the more likely your leptin circulating levels will go up. And then, of course, we also see that they develop fatty livers. And that's shown particularly in the males in the bottom left-hand side. Now, I'll say, and I'll show you more data, that the males have a phenotype that's worse than the females. And we know one of the reasons is that it's probably the ovary and the estrogens coming from the ovary that is protecting the females. Because when we take the ovaries out of the female mice, they look just like the male mice. They're more obese, more severe, and have fatty livers. So we're going to look at the male mice particularly. And this just shows that the male mice have a slight rise in glucose, but a tremendous rise in insulin. And for the people involved in metabolic disorders, you know that if your insulin's high, but your sugars are fairly normal, it's because you're very insulin resistant. And that's typical of obesity. And you can see it's more in the males on the left than in the females on the right. Well, we can prove that it's insulin insensitivity or insulin resistance in the males because if you look at the green line, this is where you inject insulin and you watch the blood sugar drop. The green line, you'll see the blood sugar doesn't drop as much as in the normals, in this blue color, or turquoise, or even the females, who seem to be quite sensitive, shown in red. And when you do a glucose tolerance test, the blue again is the glucose rising and falling rapidly in the normals, rising a little higher in the red, but falling nicely in the females, rising higher in the males and not falling. So the males don't have the ability to bring down that insulin. They're insulin resistant. And that's typical of obesity. All right. What about the tumors? We take cells, either colonic cancer cells, or lung cancer cells. We inject them subcutaneously, and we watch how many mice develop tumors and how fast they grow, to what extent. And that's shown here. That's the slide, sorry. And what you'll see in the black and the green are the regular chow male and female. 50% of them develop tumors, so 50% are tumor-free. In the red and blue are the high-fat diet, and you'll see 100% develop the tumors because 0% free at the end of the couple of weeks. That means that more mice who are obese will develop the tumors, and then the next part shows that the tumors are much, much larger in the obese animals. So if you look, for example, at the red compared to the black, these are the females, and the blue compared to the green, the obese individual mice, the tumors are enormous. And in fact, higher in the males than in the females. So this particular study was published a couple of years ago, and what it said is that it's true. Obesity, we can show in mice, the same as in humans, that cancer will be more common and will grow much faster. But the question is why? We postulated that their glucose levels are relatively normal. Their lipid levels are relatively normal. We don't know much about their inflammatory cytokines, but those are 3 of the 4 factors that could be playing a role. But the factor that we chose to focus on was the hyperinsulinemia. And we postulated that the hyperinsulinemia is acting as a growth factor. It's normally a metabolic factor, hormone, but now it's acting as a growth factor stimulating tumor growth in the obese setting. So we turn next to, what about diabetes? Can we show the same thing? Well, let me take you back 100 years. 100 years ago, this guy Maynard published that there's an association between cancer and diabetes. Both diseases have very much the same distribution in the age. And if you jump to number 5, he would say that if there were a common factor in the causation of the dual increase, a correlation between these diseases may be discovered. So what he was really saying is we'd find the cause for why there's an association between cancer and diabetes. He said this 100 years ago. And it's now 100 years later, and we still don't know the answer. Maybe we'll know the answer by the end of today. The other point that he made was that actually suicides are also increased with cancers, and that's not surprising. And he also thought that maybe it's increased with diabetics, and we know that diabetics do get depressed, not surprisingly. But the causation and the cause between the connection between the two was thought about 100 years ago, but still only until recently has it resurfaced. Let me show you a different study by another group where they looked at the relationship between diabetes and cancer mortality. And again, it's risk and mortality. And every time I show this slide, people will say to me, those who know, that most of the diabetic patients in the US, type 2, actually are obese. So how can you separate out that causation of obesity from type 2 diabetes? Maybe all of the causation is just due to obesity and diabetes is not a factor. So there you have to rely on the statisticians. The statisticians can do, with these large cohorts, they can remove the BMI as a confounding factor. And you're left just with diabetes unrelated to age, BMI, etc. And what they show is on this slide, all those cancers you saw before have an increased risk and mortality if you're a diabetic. This is in the males, and this is in the females. The numbers are slightly lower. So obesity causes a high incidence of mortality. Diabetes causes an intermediate increase. But of course, when you put the two together, you get a much more increased risk of cancer and mortality. And why is this important today? Because, again, maybe not in Israel, but certainly in the US and certainly in Southeast Asia and even African countries, there's an obesity epidemic and there's a type 2 diabetes epidemic. So no matter what we do in treating cancer, we're going to get more and more cancers because of this epidemic if all of this data is correct. And so we turned then to a model of type 2 diabetes to see whether we could look at this question of hyperinsulinemia. Just to mention, on the right-hand side, you'll see hyperinsulinemia, glycemia, hyperlipidemia. On the left-hand side, the cytokines, the leptin, adiponectin, my second favorite or first favorite hormone, IGF-1. There are a lot of possibilities, but I'm going to choose hyperinsulinemia as today's factor driving cancer. So first I need to show you about a model of type 2 diabetes to convince you that this is a good model to use in the studies that I'm going to show you on cancer. And to remind the people not studying metabolism or diabetes, diabetes is a dual defect disease. On the one side, you have to have insulin resistance, shown here, which means the tissues don't respond to insulin. Like I showed you, when you inject the mice, the blood sugars don't drop because insulin is unable to work on the liver, muscle, fat, because those are the metabolic tissues that normally respond and are defective in obesity and type 2 diabetes. But not all obese individuals become diabetic. And the reason for that is they have good genes in their beta cell. But if the beta cell is genetically programmed to become dysfunctional when put under pressure, like when you add the insulin resistance, then you can become a type 2 diabetic. So you've got to have dual defect disease. It's genetic with a lot of environmental influence, like weight, inactivity, aging. All these factors will add pressure onto the genes. You have the dual defect disease. You go through a phase of prediabetes where you can only just find an impairment in glucose tolerance. They're not grossly diabetic, but eventually they will become diabetic if all those factors play into this paradigm. So I want you to remember that there's a prediabetes and a diabetes phase, and that insulin resistance is very important and beta cell dysfunction is very important. Now, I've simplified it because we know that there are many other organs involved, the brain's involved, the gut's involved, but let's just keep it simple, otherwise I'll never be able to get back to Tel Aviv this afternoon. Okay. So we created a mouse model where we caused muscle insulin resistance. That was the primary defect in these mice, and we did it by making a transgenic animal where we overexpressed a defective IGF-1 receptor. Now, the IGF and insulin receptors are oligomers. So if you overexpress one half that is defective, it will inhibit the endogenous receptor. So it's called dominant negative because it's inhibiting the endogenous receptor. Obviously, you have to overexpress this defective receptor, but also will oligomerize with the insulin receptor because they're very similar. And so you'll knock out the function of the IGF receptor and the insulin receptor, and you won't be able to stimulate glucose uptake into muscle. So they will develop severe insulin resistance the day they're born. And that's what we found. So we've engineered severe muscle resistance. About 2 weeks later in the mice, we find that the fat cell is now resistant to insulin, secondarily, and becomes lipolytic. Normally, insulin stores fat in the fat cell. If you're insulin resistant in the fat cell, you'll break down your lipids, and out will come fatty acids. And eventually, it will lead to high triglycerides in the bloodstream, and eventually lead to triglyceride storage in muscle and liver, and interfere with insulin action. So a vicious cycle gets set up, to the degree that at about 4 weeks of age, using clamp studies, we could actually show that they were now liver resistant to insulin. Insulin was unable to suppress the liver production of glucose. And so you had high fasting glucose, and you couldn't stimulate glucose into the muscle, and you kept having lipolysis. And all of this fed into a worsening of the situation. But at this stage, their beta cell overcame the resistance by putting out more insulin. So they had high insulins and relatively normal glucose. They were insulin resistant. And at about 8 weeks, the beta cell became dysfunctional, and their blood sugars just took off, 500, 4 times the normal range. So they went from pre-diabetes to diabetes, but it all started with muscle insulin resistance. Now, my diabetic colleagues don't like this model. They say, but your mice are non-obese. And that doesn't fit the typical type 2 diabetic in North America. On the other hand, there's a positive spin. The positive spin is they're not obese. So I can take these mice who are hyperinsulinemic without obesity and see whether that drives cancer, because now I won't have the obesity as a confounding variable. So that's what we did. And while the diabetic community didn't like it so much, the cancer community thought it was a great idea. Okay, so just to show you, when they're born, they have very high insulin levels because of the insulin resistance, and that's in purple. In the blue, you'll see at the bottom the normal insulin level. So these are the engineered mice. These are the normal mice. You can see at 2 weeks, they already have high insulin levels. And at the time that they have high insulin levels at 2 weeks, their blood sugars are normal. It's only at 6 to 8 weeks that they have very high blood sugar levels. So they're pre-diabetic, and then they become diabetic. When you do a glucose tolerance test, you'll see that here their fasting sugars are normal, shown here. You give them glucose. There's a mild rise in glucose, but it comes down reasonably well. But when you look at them at 8 weeks, they're grossly diabetic, high fasting levels, and then the blood sugar stays very high. So that's the pre-diabetes moving to diabetes. What I've shown you to now with this situation is that at 8 weeks, if you look at their beta cell, so this is the normal beta cell, low fasting, you're perfusing the pancreas with glucose, low glucose, high glucose, insulin comes out. First phase, second phase. And when you look at the transgenic mice, they obviously have a high insulin, as I showed you before. They don't really have a first phase. It's kind of missing. They just go into this exaggerated second phase. And this is the time when they become diabetic. And this replicates what happens in obese and type 2 individuals. They lose this first phase. So we think, can't prove it, that loss of first phase is the timing for the development of diabetes. When we treat the diabetes, the first phase comes back. So we think at least temporally it's related. Now, what I've shown you is this scheme that we have, non-obese type 2 diabetics, but this is the male animal. And remember, I told you early on with the obese situation, the male animals usually show a more severe phenotype. We're going to work in breast cancer, so we chose to look at the female mice, of course. And what you'll notice in the female mice is that their blood sugar is normal. They have mild glucose intolerance. Their lipid levels are mildly abnormal compared to the male, but they have the same degree of insulin resistance and hyperinsulinemia. So in a way, the hyperinsulinemia is the strongest aspect of the female mouse, not the diabetes and not the metabolic aspects of diabetes. And so we thought, well, this would be a nice start to the experiment and look at hyperinsulinemia and breast cancer in the female mouse. Okay. What I'm going to show you is that our theory is as follows, that the female mice have insulin resistance. That leads to the high insulin in the circulation, and that interacts with receptors, insulin or IGF receptors on the mammary gland, and that when you add a cancer to that mammary gland, it takes off. And that the insulin working through this group of receptors is driving proliferation of the cancer. Not causing the cancer, but driving the proliferation of the cancer. For those in the field, we can't exclude that IGF-1 could be the mediator, because we know that when you raise insulin levels, you drop the binding proteins, you have more free IGF-1. So it could be IGF-1 that's secondarily involved. But we think we can exclude that with the experiments that I'm going to show you. What I need to show you is what the insulin and IGF receptors look like. This is the insulin receptor, this is the IGF receptor. As I told you, they're oligomers, and that's why if you overexpress one half of that defective receptor, you tie up the endogenous receptors, they cannot work. But this is the insulin, that's the IGF receptor. This is the tyrosine kinase. This is the enzymatic portion of the receptor in the cell. Once the ligand, in this case insulin, binds the insulin receptor, activates its tyrosine kinase, results in metabolic effects. The normal function for insulin is on the liver, the muscle, and the fat cell to keep the blood sugar normal, to keep protein in the muscle, and to keep fat in the fat cell. This is the IGF receptor, which is very similar. It's a family of receptors, tyrosine kinase. When IGFs bind this receptor, their major function is growth, development, proliferation, and preventing cells from dying from apoptosis. So it's a mitogenic type receptor. This is a metabolic receptor. These two tyrosine kinases are from the same family. They're very similar. So we don't quite yet understand how come when you activate this one you get a mitogenic effect, and when you activate this one you get a metabolic effect. But that's a horse of a different color. Okay. What we learned about 10 years ago is life's not so simple. The insulin receptor is not just a metabolic receptor, because there are two types of insulin receptor. Again, discovered 15 years ago, studied 12 years ago, and buried, and now has resurfaced in the cancer field. There are two receptors. One's called IRA, one's called IRB. And the difference between them is just a splicing out of exon 11. It's an in-frame splice, so you get exactly the same receptor, except this one has exon 11 and this one misses exon 11. But it changes the receptor, because IRA has a major effect on mitogenesis. IRB is the classic metabolic receptor. So if you look at cells or tissues, you'll find IRB on muscle, liver, and fat. And if you look for IRA, you'll find it in the fetus, where insulin is known to be a growth factor. And later on, you'll find it in cancer cells, because it binds insulin and IGF-2 very strongly, and that stimulates this receptor to cause mitogenesis. So it's possible that in this whole story, IRA is being activated by the hyperinsulinemia and driving some of the cancer that I'm describing. Alternatively, it could be insulin via IGF driving the IGF receptor. Okay? A lot of these possibilities, but what I'm going to show you is that it's insulin and IRA that are playing a role in the particular cancer that I'm going to describe. Okay, let's look at the mammary gland of our mice. These are the wild-type mice. These are the hyperinsulinemic female mice. This is the mammary gland. This is a lymph node. These are the lateral branches and the terminal end buds. But you can see that in the hyperinsulinemic mice, there's marked early differentiation of the mammary epithelium, lateral, terminal end buds. It's much more dense in these mice than in these mice. So the hyperinsulinemia is actually driving mammary gland development much earlier and more extensive in the hyperinsulinemia situation. Furthermore, which are the receptors that are doing this? This is the IGF receptor, the insulin receptor, and hybrids. This is a common IGF insulin half in each receptor. What you'll see here is that there are more insulin receptors in these mammary glands. This is in the epithelium itself, the epithelial cells. When you look at the hyperinsulinemic mice, an enormous increase in the insulin receptor. So the insulin receptor is driving this mammary gland development. Furthermore, when we look at, this is by Western blots and ELISA assays, when we look at which receptors here, half of them are IRA and half are IRB. Half of these are IRA and half are IRB. So there's a lot more IRA and IRB in these hyperinsulinemic mice, which we believe is part of the process that's being driven. Okay, on that background, we introduced cancer. And we did this in about 4 or 5 different cancers. I'll show you only a couple of them. But our first example was this oncogene called polyomavirus middle T antigen. Very powerful oncogene. When it's expressed in any tissue or any cell, 100% of those animals develop cancer. So we expressed it in the mammary gland because that's what we were interested in. We used a promoter called the MMTV. It's a mouse mammary tumor virus promoter, which would drive the polyomavirus expression only in the mammary gland. And we expect to see 100% tumors in the mammary gland of mice. Now, the polyomavirus is a transmembrane virus. It has tyrosine and serine, which become phosphorylated, the amino acids. And these are the substrates that are commonly drawn to the polyomavirus, activated by activating this virus, and then turning on this very strong oncogene. The oncogene itself doesn't have an enzyme, so it's got to be phosphorylated by some other enzyme. And then phosphorylated draws these substrates, and the oncogenic process goes on. So what we did is we took mice that were expressing the polyomavirus in the mammary gland specifically, and we crossed it with our hyperinsulinemic female mice. So we got this double transgenic. The idea was to see all these mice would develop tumors, but what would happen in the face of hyperinsulinemia? Here's the tumor, or the preneoplastic tissue, preneoplastic focus that develops in all the polyomaviruses, the mice with polyomavirus at 6 weeks, very early on. And you'll see it's in one zone of the mammary gland. Whereas when they have hyperinsulinemia, you can see this is the primary focus, but there are multiple secondary foci, so it's spreading much faster in the face of hyperinsulinemia. And if we wait 2 or 3 more weeks, these tumors that develop infiltrate the chest wall. So it's a very aggressive tumor, especially when you have hyperinsulinemia. It's so aggressive that we couldn't use these mice, because what we wanted to do was to manipulate the mice for about 3 or 4 weeks with something against insulin, something against the receptors, to play with the mice, give them various compounds. But what you can see is this rapid onset and aggression was basically didn't give us a window of opportunity to study the mice. So what we did, like many other groups do, is you take cell lines that are derived from these tumors. You use the cell lines to inject into the mice at different ages, and you can follow the tumors. You can make smaller tumors, larger tumors, depending on how many cells you inject. And basically, we got the result whereby you take the cell lines, you inject them into wild-type normal mice or hyperinsulinemic mice into the mammary gland, and you'll see that the tumors, after 4 weeks post-injection, grow to a very large size. These are the control mice, and these are the hyperinsulinemic mice. So it replicates exactly what we saw in the transgenic cross. And here you can see, again, the tumor volume at the end of the experiment, and you can see the hyperinsulinemia in those mice. So we believe that the hyperinsulinemia is driving the growth of these tumor cells, not causing it. It's caused by the polyomavirus. The question that came up in the minds of the oncologists was, well, we're not quite sure what you're telling us, because we believe, the oncologist speaking now, we believe that the polyomavirus, because it's so strong, is phosphorylated and hijacks basically all the substrates in that particular cell to set up this oncogenic process. And you're suggesting that when you stimulate the insulin or IGF receptor, which uses similar substrates, you can now add to that process. So they forced me to do some studies, which I'll show you, to show that in cell lines, which we used, as well as in the tumors that were produced, that indeed the hyperinsulinemia is driving these receptors to further phosphorylate the polyomavirus, to further set up an exaggerated oncogenic response. And basically, we turned to the cell lines. You can see some of the cell lines we used, 2 of which had the polyomavirus, both on RNA and protein. All the cell lines had the IGF-1 receptor and the insulin receptor. They're all an IRS-1 and IRS-2. These are the downstream substrates, the early downstream substrates. What you can see on this slide is the first lane is in the absence of insulin or IGF. the second lane in the presence of insulin, the third lane in the presence of IGF-1 and IGF-2. You can see here that the insulin receptor, in the absence or presence of insulin, gets phosphorylated, which is the first thing that happens to the receptor, tyrosine phosphorylation. The same for the IGF receptor, absence or presence of IGF, activation of the IGF receptor. And in both cases, you can see the absence of insulin, the presence of these ligands, you can see phosphorylation of, say, IRS1, the insulin receptor substrate number 1, which is the very first substrate. So our cell lines, and we showed in the tumors as well, were being activated by our hormones and by the receptors. But now what we had to do was to show that this activation was also capable of further activating this process. To do that, we decided to show that maybe the two interact with each other, that when we stimulate these receptors, they bind the polyomavirus. They've got to get in close proximity in order to phosphorylate. So that was done by co-immunoprecipitation after activation. And I'll just show you a small part of it. If you look here, this is IGF-1, the absence or presence. You can see an increase. What you're looking at is immunoprecipitation of the IGF receptor. So we bring down the IGF receptor, and we look for the polyomavirus. And we show that in the absence, or in the basal state, there's some association, but it's markedly increased when you add IGF-1. Similarly, at the bottom, when you add insulin, you see an increase in the association of the polyomavirus, the phosphorylation, when you immunoprecipitate with the insulin receptor, absence and presence. So in this situation, we showed increased association, and then we showed increased phosphorylation after stimulation, increased phosphorylation, and then more substrates binding to the polyomavirus. So we were able to activate further the polyomavirus in the in vivo situation, and then set up further cell proliferation, anti-apoptosis, and invasion. Okay. So at that stage, we were able to say that yes, in vitro and in vivo, in the cell culture and in the tumors, insulin's capable of doing this, the hyperinsulinemia. Whether it's working through the insulin receptor or the IGF receptor, what you'll see is we don't know, and that's why we basically link the two together. We're not sure yet which receptor is being worked on, but we think it's the insulin receptor. And we think, because we have evidence, I showed you the mammary gland, but we also have in cell lines and in the tumors, they do express IRA to a significant amount. So this is our theory so far. Insulin resistance in the female mice leading to hyperinsulinemia, stimulating perhaps the insulin receptor, but maybe the IGF receptor, activating downstream pathways, which will then further activate this polyomavirus effect. And we showed it with the MAC-new oncogene, which is the ERB. We showed it with chemical oncogenesis. So we've shown it with 3 or 4 cell lines and types of tumors, and we get the same result. Well, to prove this, we said, if we block the insulin receptor and the IGF receptor, we should be able to prevent this function. You know, it is possible that glucose is doing it, that cytokines are doing it, but if it's insulin, we should be able to show that by blocking the receptor. And what we did was, we got a tyrosine kinase inhibitor. Remember, the enzymatic process occurring in the activation of the receptors and the downstream function is tyrosine kinase. Today, there are about a dozen companies that are working on looking at blocking the IGF receptor to treat cancer. Some of them are making antibodies to the IGF receptor, and some of them are making tyrosine kinase inhibitors, which are small molecules. So we got this tyrosine kinase inhibitor from Bristol Myers Squibb. You know, in the US, you always have to be transparent and tell who you spoke for, who you advised, and where you got the compound from. This is from Bristol Myers Squibb. This tyrosine kinase inhibitor, originally developed against the IGF receptor, had a disadvantage. The disadvantage was it also inactivated the insulin receptor, because the two kinases are so similar. But they are different from every other kinase you know, so they're very specific. But they are very specific for the two receptors. That's the downside. The positive side is it blocked both receptors. So at least for our initial experiment, we didn't care, because we're still not sure which receptor it is. And what we found was, When we gave this blocking agent, this tyrosine kinase inhibitor, to the mice, their hyperinsulinemia got worse. We weren't surprised. You'll see here. This is the hyperinsulinemia in our regular female mice, and this is the hyperinsulinemia once you've given this tyrosine kinase inhibitor, because you're blocking the insulin receptor throughout the body. So you're making insulin resistance even worse. The blood sugar, which was mildly elevated, got a little bit higher, and the glucose tolerance was a little worse. But really, the major feature was the hyperinsulinemia. So in the face of this exaggerated hyperinsulinemia, let's look at what happened to the tumors. This is the tumor in the wild type. This is the tumor in the hyperinsulinemic mice. Now you give the tyrosine kinase inhibitor, you push up insulin further, but you block the receptors, and the tumor growth comes down to the regular level. So the added effect of insulin, hyperinsulinemia, is lost, and the tumors now grow just their regular size. So we believe that that proved that the insulin or IGF receptor are in fact mediating this exaggerated response in the hyperinsulinemic mice. The second to last experiment was to now ask the question, what happens if we drop the insulin levels? Will that then affect tumor growth? We had shown in the male mice that we can cure the diabetes or drop the insulin resistance and insulin levels by using 3 different techniques. One was a fibrate, because the mice had a lot of lipotoxicity, a lot of lipid storage in the tissues. Another one was leptin. And the third one was this beta-3 adrenergic receptor agonist, which causes the fat cells to burn fat and therefore remove all the excess fat from the body. So this is the one we chose to use because fibrates can only be used for about a week before they cause liver damage. Leptin's not a good idea, even though it burns fat, because leptin can stimulate cancer cell growth and may be involved in the obesity story. So we chose to use this one and gave them 4 weeks of injection. And basically, what I will show you is that their insulin levels, shown here, came down markedly. So we removed the hyperinsulinemia by, of course, removing the insulin resistance. And what then happened to the tumors? You can see that the excess growth of the tumors was brought down in one cell line, and the excess growth in the tumors of the hyperinsulinemic female mice was brought down in the second cell line. One of these, I forget which one is not labeled, one of these is the polyomavirus, one of these is the MACNU-A, and a third cell line shows exactly the same. So blocking the hyperinsulinemia seemed to reduce the tumor growth and added another nail in the coffin towards suggesting that hyperinsulinemia is driving the tumors in these mice. Okay, so by reducing the insulin resistance and the hyperinsulinemia, the added growth of the tumor was abrogated. But again, I say we cannot exclude other factors because there are mild defects in other aspects of metabolism that could play a role. And this is what I'm showing here in a sort of, just before the end, the summary slide is we believe it's hyperinsulinemia, but of course there are always mild defects in this hyperglycemia, hyperlipidemia, and there are other cytokines that may play a role. But I hope that my hypothesis of hyperinsulinemia has some support from the studies that I'm showing you. So let me show you the very last study, and it's a very preliminary study. It's on metastases. Question is, you've got increased risk, increased growth, but do the cells metastasize more in the face of the hyperinsulinemia? And of course, I wouldn't bring that up if I didn't think I had support for that. Otherwise, I would just gloss over it and end the story. So we took MVT cells. It's another cell line just like the others, except it's more aggressive in that it metastasizes more readily. And it's a breast tumor that metastasizes to the lungs. And what I'll show you is that wild type, the left 6 and right side of the hyperinsulinemic female, they all or mostly develop these metastases. You can see them starting to develop in the wild type, but you can see that it's much more extensive in the hyperinsulinemic, not this piece, but the rest. The hyperinsulinemic females have much more metastases, and they're much larger. And while this is preliminary data, we've been able to show this on a couple of similar studies. And what we're doing now is basically transfecting luciferase into these cells so we can actually monitor it by in vivo, in vivo imaging to actually see the development and the growth of these luciferase-expressing cell lines. And our preliminary data suggest that it looks just like this, more aggressive metastases in the hyperinsulinemic female mice. So I want to finish there by suggesting that hyperinsulinemia in the diabetic and obese state separately could be driving, this is endogenous hyperinsulinemia, could be driving tumor growth, but obviously we need a lot more information before we can be 100% sure. Thank you very much.